Welcome to my video on exit-based physiology. I will be focusing on FRCA primary syllabus. If you haven't watched the video on renal physiology, please click the link above and watch it before you continue watching this one. I've explained the reason why I would prepare my revision as if I were preparing the SOE, and also some skills of answering SOE. So here are some hot questions that has appeared in the past paper or in exam books. Let's go through them one by one and let's see how to deliver an answer beautifully. So these are the commonly asked definitions, and unfortunately you have to memorize them by heart. And also remember the standard conditions for this definition, like the body temperature and the standard partial pressure of carbon dioxide. I'm not going through these details here. So explain the Henderson-Hasselbalch equation and its clinical significance. This is the kind of question that you have to learn how to derive an equation. Practice more outlining the equation so you can be slick in exam. The Henderson-Hasselbalch equation is an equation that relates the concentration of a weak acid and that of its base in a solution. The Le Chatelier's principle, if that's how it is pronounced, states that the hydrogen ions are added to the solution, the equilibrium shifts to the left, and the hydrogen ions will be neutralized by the conjugate base to minimize the increase in the free hydrogen and maintain a constant pH. And by the law of mass action, the equilibrium constant K, relative concentration of the acid and the conjugate base, together determines where the equilibrium is. The clinical significance of the equation is that by measuring the pH of the solution, the concentration of acids and conjugate base, we can derive the pKa of that weak acid or weak base. By knowing the acid or base nature of a drug and its pKa, we can compare the speed of onset of action. For bases, the lower the pKa, the weaker the acid is, and the less ionized it is at physiological pH. Acids are reverse, and the higher the pKa, the weaker the acid is, and the more unionized it is. Local anesthetics are commonly used anesthetic drugs for comparison. The pKa of lidocaine is 7.9, while it is 8.1 for bupivacaine. Therefore, when local anesthetics, which are weak bases, are introduced to our physiological pH of 7.4, which is below their pKa, they are only partially unionized. The proportion of unionized drug is more the lower the pKa. Therefore, lidocaine has a higher proportion of unionized drug at physiological pH than the bupivacaine. This influences the speed of onset of action, as it is unionized drug that diffuses across the phospholipid membrane. The drug with lower pKa have a quicker onset of action. So the next question, how does our body deal with acid in blood? The mechanism can be broadly divided into buffer system, which acts in seconds, respiratory responses, which acts in minutes, and renal response, which takes hours. Again, try to structure your answer and give headings. The buffer system can be further divided into extracellular and intracellular buffers. The main extracellular buffer is the bicarbonate carbonic acid systems, followed by hemoglobin and plasma protein. Main buffer intracellularly is the phosphate, followed by cellular protein. Each of these buffer systems has their capacity, depends on their pKa and prevalence. An ideal buffer should have a pKa close to body pH, making it half-half dissociated, and a large capacity. So bicarbonate is the most important system. Despite it having a low pKa, 6.1, relative to the blood pH, it is effective due to the ready excretion of carbonic acid in the form of CO2 by the lungs, and the continuous regeneration of bicarbonate in the kidneys. It is more efficient at buffering exits since its efficiency increases as the pH falls. It is the main buffer system in the blood due to abundance of plasma bicarbonate. The production of carbonic acid is catalyzed by the enzyme carbonic anhydrase, which is present in the red blood cells, but not in plasma. The diagram on the right shows its greatest buffering capacity at a pH 6.1, resisting a change in the pH. Hemoglobin has a histidine residue that can dissociate and buffer. Oxyhemoglobin and deoxyhemoglobin has different buffering ability due to different affinity to the hydrogen ion by the Haldane effect. Deoxyhemoglobin is a better buffer with a higher pKa than the oxyhemoglobin. The protein buffer, intracellularly, and plasma proteins have carboxyl and amino groups that can dissociate. And finally, the phosphate is effective intracellularly than extracellularly. It primarily acts in the tubular fluid of the kidney, where phosphate is concentrated near its pKa, and it has a pKa of 6.8. So the respiratory system has a control effectiveness of 50 to 75%, which describes the ability of a homeostatic mechanism to the change of the parameter it controls. Therefore, the lungs will only deal with 5 to 7.5 millimole of each 10 millimole change in hydrogen ion concentration. The more hydrogen ion present, again by the Henderson-Hasselbalch equation, the more the equilibrium is shifted to the left and produce more carbon dioxide with the help of the carbonic anhydrase. The increase in the partial pressure of CO2 is sensed by the chemoreceptors and stimulate the respiratory center to increase the minute ventilation to eliminate this carbon dioxide. So how does the kidney handle exit? The kidney tubules handles exits by both hydrogen secretion, bicarbonate production, and buffering system. The kidney tubules on these electrolytes involve very complicated electrolytes exchanges, but I can hardly believe you would get drilled into such details in exam. You would be either getting a gold medal or close to being failed if you do get asked. Being said that, I do want to highlight a few points just in case, or if it appears in MCQ. I would not bother too much drawing these diagrams in exam settings. So for the reabsorption of bicarbonate, the proximal tubule accounts for 90% of all bicarbonate reabsorption, which involves carbonic anhydrase. This process also occurs in the distal tubule and the collecting duct, but only accounts for 10%. This occurs with hydrogen-potassium ATPase, and less importantly, sodium-hydrogen linked transport. What's important is that the aldosterone works here. Aldosterone increases hydrogen secretion by increasing the hydrogen ATPase in the apical membrane of the intercalated cells. It also acts on potassium channel in principal cells. It explains how aldosterone increases the hydrogen and potassium excretion. For buffering, it involves phosphate and ammonia buffer with complicated pathways. Alkaline phosphate is filtered into the tubular fluid. This dissociates, and one sodium is exchanged for a hydrogen ion to form exit phosphate to be excreted. The pKa is 6.8, and it mainly occurs in the distal tubule. For ammonia buffer, it is produced by deamination of glutamine. The ammonium is secreted as ammonia by the collecting tubule ducts and converted into ammonium in the lumen by combination with secreted hydrogen. The bottom line is, more the ammonium secretion occurring in the tubule, the greater amounts of bicarbonate will be available in the plasma. So how does the chronic renal failure affect exit-base balance? Chronic kidney disease is a progressive disease, and exit produced by metabolism will gradually increase and become more than it is excreted. Buffers will eventually be depleted. Reduced total number of functioning nephrons will reduce the production and secretion of ammonia, and therefore reduce buffering of the urinary hydrogen, and eventually reduce tubular secretion of hydrogen. Excess potassium causes intracellular alkalosis, which inhibits hydrogen secretion, and bicarbonate reabsorption and regeneration will be reduced. Hemoglobin levels are reduced due to depressed production of new red blood cells from diminished erythropoietin secretion. Plasma proteins may be diminished in the presence of increased glomerular permeability in certain conditions, like the glomerulonephritis or nephrotic syndrome. Last but not least, our liver plays a role in buffering. The liver is highly metabolic active and responsible for CO2 production from oxidation, metabolism of organic acid anions, production of plasma proteins, and metabolism of ammonia. So here's the end of exit-based physiology. Thank you for watching my video. If you haven't subscribed our FRCA primary resources platform website, please click the link below and subscribe it, so you will get notified once we have any new videos. Thank you.